the effects of thermal history on intra and transgenerational plasticity in Laminaria pallida. So firstly, a little bit about plasticity. It's a phenotypic adaptation to environmental stressors, and it's called transgenerational when it is transmitted from parents to offsprings in a non-genetic way. And this is something that is especially significant in sessile species, such as algae in my case, because the offspring are quite likely to face similar pressure as the parents and also do not have the possibility to move away. And it's important because it decreases the resilience of species to sudden or short-term stressors, and which is relevant in the context of climate change with rapid changes in ocean acidity or water temperatures in our case. So Laminaria pallida is a habitat-forming species and the dominant kelp species in Namibia and Western South Africa, with distributions ranging from Rocky Point in the north to Danger Point in the east, and it's also found in some places in the Southern Ocean. As a result, the temperature range is between about 5 to 22 degrees Celsius, but the largest beds are found south of Luderitz, which is marked on the map, where the temperatures are more mild. For our experiment, however, the samples were collected in Swakopmund, which is a bit far further north, and where the distributions and the populations are a bit more sparse. Sporophytes typically reproduce year-round, but there is a definite peak at the end of the austral summer, so around March. And gametophyte reproduction, on the other hand, is mostly linked to environmental conditions, as they require quite mild temperatures. So looking at the kelp cycle, at the life cycle of kelp, it's distinct between two phases. So first, there's the diploid phase with sporophytes, which are the large mature algae. And these sporophytes release zoospores, which are haploid and then differentiate into either male or female gametophytes. And when the conditions are right, these gametophytes undergo gametogenesis, where the females form oogonia, which then release an egg as well as pheromone. And this pheromone triggers the release of sperm from antheridia on the male gametophytes and also attract the sperm. And once these eggs are fertilized, they grow into juvenile sporophytes, which are diploid once again. So for my research, we tried to look at how long-term thermal stress in gametophytes affects their reproductive ability, as well as the heat tolerance of their offspring. And we tried to look as well as male and female effects in gametophytes and maternal or paternal effects in sporophytes, as well as potential inbreeding effects. So for the first experiment, we looked at how gametophytes respond to a long-term thermal stress. And for this, from 3 individual sporophytes, we collected both a male and a female. So in total, we had 6 strains gametophytes, and they were exposed for 3 months to 8 and 20 degrees, which are the boundary temperatures of the natural distribution, and put in red light so as to inhibit reproduction. And their physiological status was estimated by measuring the photosynthetic efficiency before and after the exposure to these experimental temperatures. So in terms of the results of this first part, here we have a graph. So on the y-axis, we have the normalized maximum photosynthetic yield, and on the x-axis, each of the 6 strains. And in blue, the bars show the experimental temperature of 8 degrees, and in red, the ones at 20 degrees. And we see that for every single strain, there is a significant difference, with the gametophytes being exposed to 8 degrees being, having a higher photosynthetic efficiency and therefore being healthier. Also, when we looked at pictures after 3 months, we can see that on the picture on the top, we see that the lattice of gametophytes that forms when they grow is quite extended, and whereas on the 20 degrees on the bottom, we see that there are bigger, thicker clusters, and we can see that quite a few cells are quite pale, which again indicates kind of a lesser health. So moving on then, we looked at how this thermal history influences the gametophyte reproduction at optimal temperature. So for this, within each thermal history group, we crossed all 6 of the strains to obtain nine crosses, of which 3 were inbred and 6 were outbred. And we placed them for 30 days in optimal reproduction conditions, so at 14 degrees Celsius, which is also the midway point between 8 and 20, and in white light. And there we measured the gametophyte growth for the first 6 days, so in theory before they become reproductive, as well as the gametogenesis over the first 28 days, and finally the recruitment. So firstly, looking at the growth, on this graph, on the y-axis, we have the absolute growth rate, and on the x-axis, each of the crosses with the panels corresponding to crosses with one female. So on the left, we have female number 1, and middle female number 3, and on the right, female number 6. And here, as well as for all the next graphs, the thermal history of 8 degrees represented in blue and of 20 degrees in red. And what we noted here is that there were no significant differences in the growth between any of the crosses. Between the thermal histories in any of the crosses, which suggests that this doesn't have much of an impact on the gametophytes once they are in reproductive conditions. However, looking at gametogenesis, this is a bit of a complex graph, but basically each clump of bars corresponds to one cross at one thermal history and the progression over the 28 days, with the 8 degrees thermal history on top and the 20 degrees on the bottom. And on the y-axis, we have the percentage of females at each of the measured ontogenic stages. So in gray, we have vegetative females. In yellow, we have females having released at least one egg, and in blue, females having released at least one sporophyte. And one thing that we can note from this graph is that when we look at crosses with the same female, so for example, in the red box on the bottom, we see very similar patterns in progression across the 3 graphs, and it's the same on the top. And from this, we could infer that there are very strong female effects rather than male effects in how the gametogenesis progresses. Then to quantitatively estimate gametogenesis, first we looked at the speed, and for this we looked on day 10 as 2 of the crosses are almost saturated already in reproductively active females, and looking at a further, they would lose some data on these crosses. So looking at this, here we have the results on day 10, with on the y-axis the percentage of reproductive females, so having at least released an egg or formed a sporophyte. And what we can see is that for the first 2 females, so a panel on the left and in the middle, actually every single cross shows a statistical difference, with the females from the 20 Celsius thermal history being more reproductive than the ones from the 8 Celsius thermal history. However, on the third female in the right panel here, we see that one of them actually has a higher reproductive number for the 8 Celsius thermal history, and the others do not show a significant difference. But all in all, we still concluded that there is a preference for a higher thermal history, although it is subjected again to female effects. Then looking at gametogenesis completion, so here we are on the day 28, so the last day at which we measured gametogenesis, and we looked only at females that have at least one sporophyte. And here the results are decisive, as every single cross has a higher percentage of successful females when coming from the 20 Celsius thermal history. From this, we can deduce that higher temperatures during the early gametophyte stages leads to faster and more successful reproduction in gametophytes. And this is both linked to seasonality, because as we said, sporophytes reproduce mostly at the end of the summer, and these gametophytes are likely to be exposed to slightly higher temperatures, but also to the stress posed by these high temperatures that commonly fastens and increases the reproduction success. Finally, looking at recruitment, so this time on the y-axis, we have the numbers of sporophytes per female. And what we realize is that once again, in female number 1, female number 3, 5 out of 6 crosses show better results when coming from a 20 Celsius thermal history. whereas on the last female, all three crosses showed no statistical difference. So once again, we have a preference for the 20 Celsius thermal history, which is subjected to female effects. Looking at how it impacts the life cycle in natural populations of gametophytes. So here we see that after being released at the end of the austral summer, the gametophytes are generally exposed to quite high temperatures, which leads to a decrease in growth and in photosynthetic efficiency. However, once the temperatures are cool enough for them to become reproductive, which is generally after 2 to 3 months, as in our experiments, the high thermal history really leads to a faster and a more successful reproduction, which suggests, well, yeah, shows strong intragenerational plasticity as well as very strong female effects. But now we wanted to look at whether these effects carry over to the next sporophyte generation. And to do so, we took 6 out of the 9 crosses, 3 inbred and 3 outbred, and we exposed them to a range of temperatures between 8 and 23 degrees, 23 being the upper survival temperature of the species, at least in Namibia, and we exposed them to these temperatures for 16 days. And there we measured the growth of the sporophytes between day 0 and day 16, as well as the changes in photosynthetic ability. So firstly, looking at the consequences on the maximum photosynthetic yield. So here on these graphs, each graph corresponds to 1 cross, with, again, female number 1 being on the left, female number 3 in the middle, and female number 6 on the right. And on the x-axis, we have the 4 experimental temperatures from 8 to 23 degrees. And on the y-axis, here we have a normalized maximum photosynthetic yield. And on the upper right corner here, we have a table of significant differences. And what we realize is that while there is not much difference at the experimental temperature of 8 degrees, as we go on to higher temperatures, actually most of the crosses showed an increased photosynthetic efficiency when coming from a higher thermal history. So this suggests that, yeah, the high thermal history leads to a higher efficiency of the photosynthetic apparatus in the sporophytes, and especially at high temperatures, which is very relevant in the complex of climate change, where, well, these algae might be, well, will be exposed to increasing temperatures. Moving on to growth. So here we have a similar graph with the absolute growth rate for the length of the sporophytes, and the rest is pretty much the same. And looking at the table of significant differences, we realized that there are actually some crosses that show a higher growth and increased growth when coming from a lower thermal history this time, which is quite surprising considering the previous results. But looking at the graphs, we realized that actually most of these come from female 1, where in both crosses and at all experimental temperatures, there's always this 8 Celsius thermal history leading to an increased growth. So again, the low parental thermal history leads to the increased growth in the sporophytes, but this is highly dependent on female effects, as we see that, for example, in this cross here, there's actually an increased growth at 8 degrees, quite a significant increase in growth when coming from a higher thermal history. And we obtained actually identical results when looking at the growth width of these sporophytes. So to put this back into the consequences for the life cycle, here, so once we have these intragenerational plasticity effects in the gametophytes, moving on to the sporophytes, we saw that the high thermal history did increase the photosynthetic efficiency of these young juvenile sporophytes, but however led to a decrease in growth, albeit only in some crosses. And so we can conclude that there is definitely transgenerational plasticity happening here with some maternal effects. Another thing that I didn't mention is that we didn't mention inbreeding effects because we did not find any significant results. And actually, another study by our research group found that there are very high inbreeding rates in this population due to the sparseness of populations in this area. So this might explain the lack of differences. And here are a few acknowledgements. Thank you for listening to me.